What's up, everybody? Mr. Lane is back, and again, I have taped my camera to the ceiling so that you guys can get the nice overhead shot here. Today, we're going to talk about probability. To review real quick before we talk about probability, let's go back and talk about meiosis, or gamete formation. We want to make some eggs and sperm. Remember, the goal of meiosis was to separate homologous pairs of chromosomes, that we were separating and reducing the number of chromosomes by half. So if we started with 2 chromosomes, we'd want to have 1 chromosome in each of our egg and sperm. That's going to be important because that's what we're going to use this probability square right here to do. And here we go. Let's look a little zoomed in here on one of these chromosomes. I want you to remember that a chromosome is a series of genes, so each one of these little links in here you can think of as a separate gene. Let's take this gene for right here. Let's zoom in on that. And if we zoomed in far enough, what we would see is a DNA sequence. In this case, the DNA sequence is CGCT. So this gene right here has this blue allele. But maybe there's a different allele out there. Let's look at this red one here. And this red one, if I zoom in on that same one, it has the sequence CTCT. So it is a little bit different. So now I have 2 different alleles. They code for the same gene, but they are 2 different versions of that gene. Now, where does probability come into this? We know that during meiosis, when those homologous pairs are separated, there is no rhyme or reason. That blue has the same probability of going left as it does going right, that it is random. So what that means is when you make egg or sperm, there's a 50-50 chance you're going to get either half of that homologous pair. For example, let's look at this individual up here. This individual had the same allele on both of its homologous pairs. That means whether it produces this, we're going to say this is the male up here, this sperm, or this sperm, it has a 100% chance of passing on a blue allele. Because it was homozygous, which means it had the same allele on both of its homologous chromosomes, it has a 100% chance. Let's look over here on the female. If I do the same thing here, we see again, we have a 100% chance of passing on the blue allele. If this female and this male were to mate, they would have a 100% chance of passing on that blue allele. Because if we take these 2, there's one of our offspring. Here's another one of our offspring. Or it could be this one, okay? Or it could be this one. So these squares represent the offspring, the probability of producing a particular offspring. So if they are both homozygous, we know then that the offspring have to be homozygous just like their parents. Cool. Let's take a look at another example here. What if I had the female that was homozygous but for a different allele? The male is still homozygous for blue, but now the female is homozygous for red. Well, we can see that the probability of this offspring, boom, we're going to be heterozygous. But we can also see that it's also going to be heterozygous here. We can see that it's going to be heterozygous here. And lo and behold, it is heterozygous here as well. So what we have is 100% chance of being heterozygous, which means the offspring will have 1 copy of each. Now, I'm not going to say dominant or recessive or get into phenotypes here, because this could be codominance, this could be incomplete dominance, this could be multiple alleles. Maybe I have 1,000 different alleles. It could get crazy in there. All right, let's take a look at another example here. Let's say they are both heterozygotes. So now I have 2 heterozygotes. Well, what is this going to be? So our first example, we looked at 2 homozygotes. The second example, we looked at 2 different homozygotes. Now we're looking at 2 heterozygotes, which means that this offspring has a 50% chance of this individual, the female, has a 50% chance of passing on the blue and a 50% chance of passing on the red allele. And likewise for the male, 50% blue and 50% red. Let's see what happens. Well, we know that if it's a 50% chance here and a 50% chance here, or a 1/2 and a 1/2 chance, 1/2 times 1/2 is 1/4. So we have a 1/4 chance of producing homozygous blue. Whereas we also have a 1/2 chance and a 1/2 chance That gives us 1/2 times 1/2, 2 times 2, so 1/4, a 1/4 chance of producing homozygous red. Now let's look at this in terms of probability from these two segments. So we know that we have a 1/2 chance of blue here and a 1/2 chance of blue here. We know that will be 1/4. But we have to add that because it could also be the blue coming from here and the red coming from here. That's 1/4. So we take that 1/2 times 1/2 to give us a quarter here, and then we add that to this quarter here. And 1/4 plus 1/4 gives us 2 out of 4, or you bet 1/2. So let's go back and very quickly show you what I want you to think about. Let's go back and look at one of those other examples. Let's boom, boom, and boom, boom. So let's go back and look at this example now, thinking about it in terms of probability. We have a 100% chance of passing on blue, and we have a 100% chance of passing on red. That will tell us that we have to have a 100% chance of having heterozygotes. But let's change that back and now look back at this one. If I have this one, what's the probability that I produce a heterozygote? Well, we know we have a 50% chance of passing on blue here. We have a 100% chance of passing on blue here. So we know that right off the bat, 1 times 1/2 is 1/2, and we can see those would both be homozygous. And we also have 1 times 1/2, which would give us our heterozygote. So we have a 1/2 probability of getting a heterozygous from this cross. The fun part is we spend a lot of time talking about Punnett squares, and I feel like we get all caught up in the letters, big T, little t, what begins with T, and we don't talk about the probability. I will tell you there's really 3 Punnett squares you need to know in terms of probability. This is one of them. If you have a heterozygote and a homozygote, it's going to be a 50-50. If you have the heterozygote and a heterozygote, that's going to be that 1 to 2 to 1. So you're going to have 1 blue homozygote, 1 red homozygote, and then 2 heterozygotes. And then the last one, if you really want to go there, would be this one. If you have 2 homozygotes, all of your offspring are going to be heterozygous. All of them. If you know the probabilities of those 3 squares, you can figure out many, many, many much more complex genetics problems that unfortunately we are not going to get to in this class, but they await before you. Because when we talk about multiple alleles, what if I said now I have 4 genes or 5 genes? What is the probability of getting this one individual that has 4 different genes? You can very quickly, using some simple probability, do some calculations and come up with some awesome numbers and actually do some science. So with that, I'll look up at the camera and I'll say I'll see you later. Have a good one.